Welcome back, folks, to the third part of our Unit 5 notes on heredity. The main focus of this set of notes will be non-Mendelian genetics. Um, in a future video, we'll finish up the last two chunks of these notes on environmental effects on phenotype and chromosomal inheritance, but that'll be in the next series of videos. Until then, let's get started on non-Mendelian genetics, because not everything is about you, Gregor. All right, so background, guys. I don't think you need to write down all of the history stuff. The AP Bio exam is not a history exam, but I like the context, and it also gives you a look at some of the scientific process, which the AP Bio exam does cover. So you should understand the process and the whys, okay, and the hows um, generally, but don't have to necessarily memorize every little piece of the history, like the name of the scientist who did this or the name of the scientist who did that. So, locating genes along chromosomes. Mendel's hereditary factors, which we now call genes, were purely abstract when they were first proposed. He didn't know what they were carried on. He didn't know what type of material they were. He didn't know what they were. He just knew that something in there was doing that. Today, we can show those factors are genes, and they're located on chromosomes. Cool thing here is on this image, you can see a chromosome here, and with a fluorescent dye, we can actually highlight a particular gene and where that is on the DNA, which is pretty cool. So that is the locus of that particular gene. And then cytologists worked out the process of mitosis in 1875 using microscopes. Eventually, they got to meiosis. And biologists began to see parallels between the behavior of Mendel's proposed hereditary factors and what we're actually seeing happening in meiosis, which is pretty cool. So they're actually able to connect the two and found some physical evidence for what Mendel predicted would be happening. And so that's where we came up with the chromosome theory of inheritance. Oops, sorry. So up here we can see basically what we were seeing with Mendel. We got the parent generation, yellow seeds and green wrinkled seeds, and we've got our genes and our alleles designated by R's and Y's here, capitals and lower cases. Um, and we can follow what Mendel was seeing here, where all the F1 plants are round yellow, but when we get to the bottom, we get this 9:3:3:1 ratio. And we can see how that happens by following what happens to those genes on the individual chromosomes as they go through meiosis. So we see over here the law of segregation. The two alleles for each gene get separated here as we go through, and we can wind up with a big R here and a big R there, and a little R here and a little R there. Over here, we see the law of independent assortment. So we can see blue on the left here, but the blues on the right for this chromosome pair, and it could just as easily be the other way around. And that winds up determining which way what is in the gametes when you're all done. So we can see these two laws that Mendel came up with um, demonstrated very clearly in what's happening with the chromosomes themselves. So a guy named Morgan used Drosophila melanogaster, a fruit fly, to it came up with a bunch of stuff to prove this. Okay. Some characteristics he liked about the fruit fly: they got a short generation time every 2 weeks. They produce a lot of kids, and they only have 4 pairs of chromosomes, so relatively easy to reproduce them, get a lot of them, and not have too many chromosomes to try to wade through. Okay. He noted the wild type, which is a term you should know: wild type versus normal. Wild type is the normal phenotypes that were common in the fly populations, which was, and then traits that were alternative to that are called mutant phenotypes. So wild type is normal or regular, and mutant would be something different. You can see wild type red, mutant type white eyes. So how was he able to correlate the behavior of a gene's alleles with the behavior of a chromosome pair? So in one of his experiments, Morgan's mated male fruit flies with white eyes, the mutant, with females with red eyes, the wild type. All the F1 had wild type. Cool. Okay. Dominant recessive right there. The F2 generation, then, when we made the two F1s, we get the F2, showed a 3-to-1 red to white eye ratio, but only males had the white eyes. Only males. So something was different than regular old Mendelian genetics here, where it wouldn't have made a difference whether it was a male or female. Okay. So Morgan determined that the white-eyed mutant allele must be located on the X chromosome. Morgan's findings supported the chromosome theory of inheritance. And here, you can pause the video and just work your way through this chart and see how this lines up with what he discovered. All right, so let's take a specific look at sex-linked genes then as an inheritance pattern. Unique patterns of inheritance. The first one we're going to look at, sex-linked genes. Morgan discovery with the trait correlated with the sex of flies was key to the development of the chromosome theory. It was the first clear evidence that these genes and alleles were carried on chromosomes and they were getting sorted and segregated during the meiosis process. In humans and some other animals, there is a chromosomal basis of sex determination. In humans, it's XY, which you probably know already, right? The X here is the big, large one that looks like a regular old chromosome, and the Y is this little dinky thing down here. Has hardly anything on it. Okay. If you have two X chromosomes, two of these, you'll develop biologically as a female. Whereas if you have one of each, an X and a Y, you'll develop biologically as a male. Now, in order for these to be homologs and attached to each other during meiosis and things, we do have to have some commonalities. So only the ends of the Y chromosome have regions that are homologous with the ends, corresponding regions on the X. So they are homologs. They will stick together, but they're not identical. And here we can see humans, X and Y, XX for female. So half the sperm have an X, half got a Y. Eggs, all of them have X's. So you can see the zygotes. If you got two X's, you're a female. X and a Y, you're a male. That's the XY system. There are some other systems out there, but you don't need to know those for the AP Bio test. So you already talked about the first one here. The big difference here is there's a gene on the Y chromosome called the SRY, or sex determining region, on the Y, and that's responsible for the development of testes in the embryo, and those testes produce testosterone and other androgenic hormones, which then control the development of all the other secondary sexual characteristics that differentiate a male from a female. A gene that is located on either sex chromosome is called a sex-linked gene. If they're on the Y, we call them Y-linked. Not a lot of those, because the Y is small. It's mostly junk. It's got a few of those genes that control whether or not you turn into a male as opposed to female. The genes on the X chromosome, we call them X-linked genes. And when we're talking about sex-linked genes, generally speaking, we're going to be talking about X-linked. So how are these inherited? Okay. X-linked genes follow specific patterns of inheritance. For a recessive X-linked trait to be expressed, a female needs 2 copies of the allele to be homozygous, just like anything else, right? Because you have 2 of them, it behaves like any other autosome would. A male only needs 1 copy, though. So you have an X with a little t on it for being short, let's say, and then nothing on the Y. They don't need a second one because, remember, dominant recessive, the recessive, it's kind of like code for making an inactive version of the protein generally. And the only reason why being hetero is needing 2 little t's to show up before is because if you didn't have 2, the other one was dominant. Well, if you take away no other dominant, all you got is recessive. So if all you have is recessive, you still get to do what you want on the playground, going back to our kindergarten analysis from before. So excellent. Recessive disorders are much more common in males than in females because you only need to inherit 1 recessive trait or allele, as opposed to females need to inherit 2 in order to show the recessive phenotype. So here we can see how we designate sex-linked traits and how we follow them through and draw them out for doing Punnett squares. You do need to know whether you're passing on if the trait is on an X or if the trait isn't on anything, if it's on a Y here. So we do XX for female, XY for males, and then we put the actual genotype, okay, the alleles you have for that gene, kind of as little superscripts up here. So X big N, that's a female with 2 normal dominant alleles for whatever this is. And here we have X little ny, that would be a male who shows the recessive trait, the little n for this. So when we do that, we can put the sperm on one side. So half the sperm have an X little n, half have a Y. The eggs have a big X big N, the other an X big N, because two X big N's. So what we wind up with here is, here we've got, these are females over here, XXXX. These are males over here, XYs. This is a heterozygous female and a heterozygous female, right? Big N, little n, they're going to show the dominant trait. Both the males in this case show the dominant trait because they get that from the mother only. So if you're a male, any traits that are on the X chromosome that are sex-linked traits from the X chromosome, you will inherit from your mother only. You will inherit from your mother only, not your father. You will inherit from your father the genes on the Y that, determine, testosterone levels and those types of things. But everything else that's on that sex chromosome is coming from your mom. So technically, you actually have more genes in common with your mom than you do with your dad, guys. Believe it or not. So we really are mama's boys. So that's one situation. Okay. Down here, we've got it where we've got a heterozygous female and a dominant male. In this case, the female, both of them are dominant. One is homozygous dominant, the other is heterozygous. Remember, we call this a carrier there, right? The female shows the normal trait, but could pass on the recessive trait to their kids, okay? But they show normal. And here, the male, half the males are going to be dominant, half are going to be recessive. This is that 3 dominant to 1 recessive, but only on the males that we saw from Morgan's experiments earlier with the fruit flies. Over here, same thing for the female, but if the male has X little n, all right, both the females will have an X little n. So we got one who's a carrier and one who shows the recessive trait. The only time a female can show a recessive sex-linked trait is if the dad is recessive. So dad's got to be recessive, and you got to inherit a recessive X from your mother, right here. Whereas on the other hand, the males is still 50-50. Half dominant, half recessive, because that's what the mother was. Some disorders caused by recessive alleles on the X chromosome in humans. Color blindness is mostly X-linked, we think, and that's a pretty common one, red-green color blindness. So if you have color blindness in your family, you're more likely to see it in the males than the females, because they only have to receive one recessive trait. And if you're a guy with color blindness, you definitely inherited it from your mother, not your father. Muscular dystrophy and hemophilia are also sex-linked traits, disorders. Something interesting that we see with XY organisms is X inactivation in female mammals. In mammalian females, one of the 2 X chromosomes in each cell is randomly inactivated during embryonic development. So that in every cell, you still only have, whether you're male or female, one active X gene or chromosome in every cell. Okay. The inactive X condenses into something we call the bar body. Okay. So if a female is homozygous for a trait that's on her 2 X chromosomes, it doesn't matter which X chromosomes get deactivated, you're going to show the same trait in every cell. If, on the other hand, you are heterozygous for that trait that's on your X chromosomes, it does matter which chromosome gets deactivated. Okay, so in some of your cells, you'll wind up with the dominant, if we're doing dominant recessive, allele active. In other cells, you wind up with the recessive allele being active, and you wind up with something we call a mosaic for that character. Okay, so in other words, right down here, like I said before, each female cell has just one active X, thus one active allele, just like in male cells. But those are present in different places in the body. So here we can see in the cat, depending on which one becomes inactive here, we can see this color change, right? So these cells here that have black fur, they originated from one of those early cells, early on in differentiation in the embryological stage, that came from this one. And then once that happened, all the cells that came from them, they're going to have black fur. So you get big chunks of black fur, and you can basically trace back from this pattern which individual cells in this cat came from which particular spot on the embryo, which is pretty cool. Over here, here's the orange fur. That's why you get those funky color patterns, and you only see them on female cats, not on male cats. All right. At this point, if we're in class, we'll be breaking to do some practice. If we're not in class, or you're with a sub, or you're doing this at home, what I'd like you to go to is the extra genetics problems, or not extra genetics problems, sorry. I'll change this later. I would like you to go to the pack we were working on before, and towards the bottom of it, there are a series of sex-linked problems for you to do. I'd like you to do 10 and 11 at a minimum. If you want more practice, keep going, but 10 and 11 at a minimum. And then for pedigrees, do 19 and 20. I think those are the only two pedigrees on there. And those pedigrees, hopefully you remember doing those in biology. If not, I'll help you out. I'll make a little mini video on those, or we'll talk about it in class. But for those two, you can track and figure out whether you likely have a sex-linked trait or not based on the patterns you're seeing in the pedigrees. Go ahead and do that. When you're done, check with other folks, see how you're doing, check the answer key to see if you did them well. If you have questions, come in and ask me, and I'll help you out with them later. But for now, we'll head on to the next section. All right, folks, so hopefully your practice on sex-linked traits and pedigrees went well. If you do have questions, remember, come back and we'll talk in class and we'll work through them in class together. I think it'll be easier than trying to do that over a video. So let's move on to autosomal linked genes. So these are genes on the autosome, so the non-sex genes, non-sex chromosome genes, that are something we call linked, which we'll explain here as we get through the notes. Linked genes tend to be inherited together because they are located near each other on the same chromosome. So each chromosome has hundreds or even thousands of genes on it, except for the Y. Remember, that's the little bitty dinky thing. Genes located on the same chromosome that tend to be inherited together are called linked genes. So looking at this picture down here, which set of chromosomes do you think? We got a G here and an A there. These are the two locus. And over here, we got G here and A there. Which of these two chromosome pairs do you think is more likely to have linked genes? Yeah, the one on the right. These genes are closer, so the chances of a crossing over event happening between them so that they're not inherited together are pretty small. And we can see that right here. Here's crossing over, and it shows you right there, bang, they crossed over together and they're still linked. Whereas over here, they're so far apart on the chromosome, and since chromosomes on average have a crossover event, 1 to 3 crossover events per chromosome per meiosis, these ones would generally not be linked. Could they be? Yes. But statistically, the chances of them being linked and inheriting together would be pretty small. Whereas over here, it's pretty big. So how does linkage affect inheritance? Morgan did some other experiments with fruit flies to see how linkage affects the inheritance of two characteristics. He crossed flies that differed in traits of body color and wing size. Okay, I'll let you go through and work out the system here, so you just pause it and do that. But the important thing for us is if the genes were not linked, the expected ratio of the different phenotypes he got would have been 1 to 1 to 1 to 1. If the genes were on the same chromosome, if they're tightly linked, you would have expected to see 1 to 1 to 0 to 0, but he got something different. He got basically 1 1 0 0, but he did get a few. Okay, and most of them were over here. So it tends to point towards genes on the same chromosome, but we got some crossing over, and that's what we got going on. So he found the body color and wing size are usually inherited together in specific combinations, the parental phenotypes, where they haven't crossed over. He noted these genes do not assort independently, and reasons they were on the same chromosome, because most of them are. Okay. However, non-parental phenotypes were also present. Right here, you got a few of those non-parental ones. Understanding this result involves exploring genetic recombination, which we've already talked about, the production of offspring with combinations of trait different from the parents. So, offspring with a phenotype matching one of the parental phenotypes are called parental types. The offspring with non-parental phenotypes, or new combinations of traits that weren't seen on the parent, are called recombinant or recombinant types. With that, 50% frequency of recombination is observed for any two genes on different chromosomes. If they're on different chromosomes, right, they got a 50% chance of both winding up going into the same egg or going into opposite eggs, depending on which way the chromosomes assort at um, when they're going up in metaphase. So 50%, 50-50. Gametes were yelled around. Here's a test cross, and this is the results we'll get. YY, RR, YY, RR, Y, so on. Okay, so what we'd expect. Okay, Morgan discovered that genes can be linked, but the linkage was incomplete because some recombinant phenotypes were observed. Remember, we didn't get all of them linked. There was those 200s ones we saw in there. All right. He proposed that some process must occasionally break the physical connection between genes of the same chromosome. Came up with the idea of crossing over. And you guys can just pause this part and take a look back. I think it'll be easier for you to self-analyze this to figure out what's going on. So, new combinations of alleles. Where do we get this variation for natural selection? So first off, don't forget that brand new alleles come from mutation only. Okay, but new and novel combinations of alleles and combinations of genes occur via all these processes of meiosis and recombination and fertilization that we've been talking about. Recombinant chromosomes bring alleles together in new combinations and gametes. Random fertilization increases even further the number of variations that can be produced, and that abundant genetic variation is the raw material upon which natural selection works. Quick thing here, mapping the distance between genes using recombination data. So a little bit of scientific inquiry here. Sturtevant predicted that the farther apart two genes are on the chromosome, the higher the probability that a crossover will occur. Just makes sense. If they're really close, the chances of the crossover happening right between them, pretty small. But if they're pretty far apart, there's lots of places for crossover to occur in between them, and they're more likely to not be linked. So from that, you can make a linkage map, a genetic map of a chromosome based on recombination frequencies. Distances between genes can be expressed as map units, one map unit or centimorgan. It's always nice when you have a unit of measurement named after you. I wonder what a centihamele would be. A centimorgan represents a 1% recombination frequency. Okay. So map units indicate relative distance in order, not precise locations. So the percent difference here, how far apart they are as a percent of the chromosome. Genes that are far apart on the same chromosome can have a recombination frequency near 50% or near 50%, remember, is the point at which would happen depending on which way they line up at the metaphasal plate, getting pulled to opposite sides or getting pulled to the same side. If they're far enough apart, the chances of a crossover event occurring is pretty high, and it can nearly mimic being completely unlinked. Such genes are physically linked on the same chromosome, but genetically unlinked and behave as if they're on different chromosomes. This one here, you can just kind of see some of the data that Sturtevant collected on the fruit flies and how far away they are. It's not something you need to know for AP Bio, though. So, what are some effects of physiological processes on genes? So, something else non-Mendelian here that he didn't cover. Some genes are expressed differently in different physiological environments. For example, there are things called sex influence traits, not sex linked, not traits that are on the sex chromosome, the X or the Y, but traits that are influenced by what sex you are will influence how genes on regular old autosomes are expressed. So you'll see here a trait that will display differently depending if you're male or female. And this generally relates to the effects of hormones on gene expression. We did mention this when we talked about cell communication in the last unit, how a lot of hormones will directly impact which genes start producing their products, right? Those hormones actually activate a DNA polymerase or RNA polymerase, which will open up the DNA and start making RNA to make a particular protein from a particular gene. Okay, so if you're male, you're going to be producing a lot more testosterone, which will activate genes differently than if you're a female who's producing less testosterone and or more estrogen. So a classic example is balding, though there is a little bit of research that's making a slightly less of a classic example now, but we'll use it as a thought experiment here, not a, this is exactly how balding works. If little b recessive is balding and big B is not going bald, um, if you inherit little b, both male and females, they go bald. You don't have any of the not going bald gene, right? So you're going to go bald. If you're heterozygous, only the men go bald, whereas if you're homozygous dominant, nobody goes bald. And the differences that we see here, so this we'd expect, and this we expect Mendelian wise. Here, this is different. Okay, this is different, and this is due to testosterone levels. More testosterone, you're more likely to exhibit this trait or less estrogen than if it was the other way around. Other sex influence traits that you might see, things like body hair and muscle mass. A lot of the secondary sexual characteristics are sex influence trait. Both the male and the female have the same genes to make the same parts, but because they have different levels of hormones released during key points in the development, say in the embryo or at puberty, those characteristics will either get enhanced or reduced. So, you know, more hair, more muscle mass versus breast size and things like that will get changed because of the hormone levels that are produced. Some other exceptions to Mendelian inheritance. Organelle genes. We talked about this earlier in the year. We talked about mitochondria and chloroplast. Extranuclear genes are genes found in organelles in the cytoplasm. In us, that would be things like mitochondria in plants, mitochondria and chloroplasts. Um, they'll carry small amounts of circular DNA, plasmids or plastids, like in bacteria. We talked about the endosymbiont theory before, how we got a symbiotic relationship there. Remember that? So these extranuclear genes are inherited maternally because the zygote cytoplasm comes from the egg. So you got the egg, sperm comes in and fertilizes. What you get from the sperm into this big cell, sperm is one of the small cells in the human body, egg's one of the biggest. What you get from the sperm is just the DNA. All the other stuff in there, there's not much in a sperm. It's pretty lean and mean so it can swim, is the DNA. All the other mechanisms in the cell, the cytoplasm, the ER, the ribosomes, the mitochondria, all come from what's already in this unfertilized egg from the mother. So when it starts dividing, since the mitochondria is self-replicating and isn't controlled by chromosomal or nuclear DNA, all your mitochondria come from your mother. So when people talk about a mitochondrial DNA analysis, that's really good for tracking maternal lineages. So your mother and your mother's mother and your mother's 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 mother going back in time. Can't track male lineage, but you can track your maternal lineage that way. And mitochondrial DNA doesn't mutate regularly, so it stays pretty consistent over long periods of time and many generations. Um, so here we can see, oh, I had this picture and I forgot about it. Um, so here is the oocyte, here's the sperm, okay? And when the sperm and the oocyte mix, it's the same mitochondria, no matter whether you're male or a female. All right, at this point, folks, we're going to pause. And in Classroom, there is a document called the Chi-Squared Test skills exercise. It goes along with an exercise in your book on page 302 in chapter 15. If you have your book, I'd probably just open it up and read it there. But I did take screenshots of it and put it in a document for you. The first page in that document is the whole page from the book. It's a little hard to read for me, bad eyes. So if you want to, I did make bigger versions of it down below, and it will walk you through this problem on figuring out if genes are linked or not linked and doing a chi-squared analysis of that. So it's going to be some chi-squared practice for you. And I think the exercise does a pretty good job of walking you through it. First thing you're going to do is fill out, basically finish off this chart. Okay? And then you're going to do your chi-squared analysis here. There's this chart you can fill out to help you organize your chi-squared test, and then you'll be able to see if it is statistically significant or not, the changes that are happening here. Were you able to support or reject your null hypothesis? So at this point, I would like you to go through and do that. You can do that with partners, you can do it on your own. When you are done, double check your work with your partners or with other groups to see if you guys agree on your answer. Um, then in class, if everybody got it right, we'll just move on. If you have questions, then we'll go over and dig a little deeper. So go ahead and do that now. When you're done, you can pick back up with the video. All right, folks, hopefully that chi-squared activity went well for you. Um, when you are back in class with me or I get back, depending on which way we're going here, we'll double check your answers and see if you were right. If you were, great. If not, we'll go back and figure out where we need to make some corrections. What we can see here, guys, just kind of a summary of what we've been talking about with this chromosomal patterns of inheritance. Up on top, we got a sperm and an egg from two individuals. We can see we got some genes, the D gene and the E gene here on different chromosomes, and we got big D allele there, little D allele over here, for instance. Then we can see we got this third chromosome, which has 4 different genes on the same chromosome, 3 of them close together on top, 1 of them further away down here. If those two come together, we get this is a potential F1 cell. 2n equals 6 chromosomes, and it is heterozygous for all 6 of the genes shown. Okay. Each chromosome has hundreds or thousands of genes. 4, these 4 right here, are shown on this one. The alleles of unlinked genes are either on separate chromosomes, like E and D are completely unlinked, they're in different chromosomes. They're going to show a 50-50 split on how they assort into different cells during meiosis. Others are so far apart on the same chromosome that they practically assort independently, like C and F. They're so far apart that they're going to be near, if not 50%, because of the number of crossovers that are going to occur between them. Genes on the same chromosome whose alleles are so close together that they do not assort independently, like these 3, are said to be genetically linked. All right. And that does it for 5.4, non-Mendelian genetics. Join me back in a little bit if we have time, and we'll get into 5.5, the environmental effects on phenotypes, or why does everything have to affect me so much? See you in a bit, guys.